What's for dinner? Nothing. Doesn't smell like nothing. Samsia's food. Thank you, Samsia. It looks good.
Hello.
Hello.
Hi, Julie.
Hi.
How are you? So this week.
And last week. Because it was like
Monday.
So last week, I ran that was a blog that I was supposed to run by myself, and it didn't go too well. There was a problem with one of the steps. We couldn't explain what happened. So between the whole time that I've been here, neither my mentor nor I, or my supervisor, nor I have been able to get one going all the way. But hopefully next week is going to be better. We got new antibodies, and we are actually working with new cells now, so it should be a better result now. I actually today I took the initiative to propose to the supervisor taking actually pictures of the cell cultures as the treatment is implemented, and so I did that for the first time, and it was pretty cool. I actually got the cells pretty nicely, and I will do that throughout the week until the treatment is done. And then next week I will be running, I think, 3 or 4 master blots to test the two agonists for BDNF. And the guys are laughing, so I guess they're not paying attention.
Okay, so at the beginning of the week, we came back from FENS. It was a really nice conference. Amsterdam was really great. And the day I got back that morning, it was pretty tiring because we got there we left at 6 from Amsterdam or 7. Anyways, and so I started working. Pablo had started on Monday, and then I finished a Western blot on Tuesday, which we were still doing IL-1 beta. And then we finally got our antibody for IL-6. And so on Wednesday, I started doing a Western blot, testing IL-6. And with the same tissue that we've been testing for IL-1 beta. So we're still developing our little diagram of exactly where the cytokines are localized in inflammation. And what else? There was something else. Oh yeah, so it's confirmed that Pablo's main project for the summer has been pushed back until September, which involved the rats because there was a virus, and so they're gonna have to clean out where they store them, and there's and that's going to take a long time, and so he's not going to be able to do the surgery until September, which I'll be gone. So it kind of it sucks in a way, but you know, you can't. Nothing's perfect. But that was last week.
So for the last 2 weeks, I've been focused on siRNA transfection in the lab, and actually 2 weeks ago we did a big transfection project where we transfected the cells with siRNA and waited 24 hours, 48 hours, 72 hours. So there was, a lot of samples, to say the least. So from there, that took about a week to do. And then last week, when I came back from FENS, I started a Branford protein determination, as well as Western blots for all those samples. So it was ambitious, and it went okay, but I think it's going to have to be repeated. Other than that, I had lab day out, which was a lot of fun, and it was a great bonding experience for my group, molecular genetics and cell biology. Otherwise, I'm going to repeat the siRNA transfections, Western blots, protein determinations, and that's it.
So last week, what I did mostly was to take pictures of my slides. We did quite a lot of slides in the first 2 weeks when I didn't have the microscope yet. So last week was literally me taking a lot of pictures and getting the right ones, exposure times, and the right parts to show the right nerves and the right stainings. And it took me quite a lot at the beginning because I was kind of having a hard time trying to get the nice picture for like 100 times focus. 100 times magnification. But last week I would say much more trials for me. But this week I'm more into it, and I feel like it got way better. So also last week I have my PI check the first pictures that I took, and he also told me how we're going to use it and how I'm going to make the pictures for the publication that he's working on right now. And also I assisted one of the PI, Dr. Bart, for one of the surgeries on the guinea pig, trying to measure the pressure in the bladder, which is the in vivo part of the project, which was really cool. And the weekend we were in Paris just to cool everything down from the week. And I wrote my introduction because my PI asked me to write it by Wednesday. So I have it done right now, and then he's going to check it on Wednesday and let me know what's next. So as usual, he said it was too long, and I've always been too long, so I'm going to cut it down and hopefully it goes well.
So last week we did another BRDU staining because we didn't do it correctly, so we actually got a completely different protocol, and it actually worked. So now we have to take 40 brains, and we have to double stain it with DNA methylation and BRDU to see if it's the same kind of cells that we want. And then besides that, I've been doing stereology every day and counting cells, and I'm getting pretty good at it. That's what I did actually for the entire week, was be in a dark room counting cells. That's it.